Hi students. So this is our second lecture in the uh, Biology 2 online course. I'm your instructor, George Pinchuk, and this lecture will be about the origin of life. So you see the pictures here showing different stages of forming of life, the molecules of life, conditions at which the life probably formed, first cells, etc. Let me just make a couple of remarks about the relationship between scientific study of the origin of life and religious beliefs about the origins of life. Uh, as you remember from the first lecture, science is entirely based on the scientific method, right? And the first step in the scientific method is observation. Observation means using your sensual organs, your eyes, your ears, your nose, your skin for the sense of touch, etc. Uh, there are certain things that are unobservable. Most religious revelations imply God or gods, divinities that are invisible. Uh, we believe that you may believe that they exist and that they do something, but you cannot say, well, I will build a better telescope and it will allow me to count fingers on God's hands or something like that. That would be demeaning even for religion, not to mention for science. Here, there are some pretty pictures of how different religions envision the creation of the world and life. This is ancient Egyptian, this is Shinto Japanese, this is Judeo-Christian, this is Hindu picture, and this is again Christian uh, picture of God creating various kinds of animals and humans. And again, don't take it wrong. Some people say that you can be either a scientist or a believer in God. That's just totally untrue. You may believe in all these revelations or in some of these revelations, but you just cannot make them the object of science. Right? You can make only observable things uh, an object of science. So let's imagine that we are scientists and we want to address the issue of uh, the forming of life on Earth. How, and we can ask this legitimate question: How could life develop on our planet? See, we're not asking questions like, what's the meaning of life, right? Or who is the creator of life and uh, where he is and what or they are, etc. Those questions are outside of the scope of science. But we can ask, on the other hand, how could life develop? It's purely mechanically. We're asking this kind of like a person who is examining the work of his or her car, opens the engine and looks at this engine and figures out how the parts fit together, how the engine starts, and how it works. So similarly, we can ask this legitimate scientific question: How could life develop? Uh, of course, we cannot go back in time and observe directly how life developed on our planet, but we can hypothesize, we can advance hypotheses based on some research done in other areas, not biology, but areas like astronomy, astrophysics, etc. And this research implies first bullet point, that the planet Earth was formed about 4.6 billion years ago and was so hot that it remained molten, thus excluding the existence of life as we know it. We cannot imagine life on a surface which has the temperature of many thousand degrees, right? No cell will maintain its structure. No biochemical reaction will proceed. So the moment or the point of time when the Earth appeared as the planet was not conducive to life, was not compatible with life. But then the Earth gradually cooled. The early Earth atmosphere, if we can use this term because it was pretty primitive, just collection of substances. This early atmosphere contained water vapor, and when the Earth gradually cooled, liquid water formed. The vapor condensed into water. Again, these are data from other areas, especially astrophysics, studying planets that are forming like right now, and kind of exporting this knowledge or applying this knowledge to our planet, hypothesizing that that was how it was. So liquid water formed, and it formed in a huge quantity so that oceans of water appeared, and there is this notion that the early Earth had its entire surface was covered with oceanic water. Where did water come from? Where did other ingredients of this primitive Earth atmosphere, where did they come from? It's not exactly known, but one pretty popular hypothesis is that small molecules, like molecules of hydrogen, molecules of water, molecules of ammonia, molecules of methane. Methane is the sort of the first organic molecule, the smallest of all organic molecules. They are extraterrestrial. They were brought to the planet Earth by asteroids, meteorites, something coming from other parts of space, quite possibly from other galaxies. And so these elements, these little molecules just remained in the Earth atmosphere. And when, again, when the cooling started, what liquid water appeared. That was the of utmost importance, because life as we know it requires liquid water, right? There are no forms of life which would maintain metabolism, maintain activity, and be completely devoid of molecules of water, liquid water in their cells. So the cells must have water. Then there were two scientists in working at the University of Texas of Chicago, Urey and Miller. In 1953, they asked this question: is it possible to simulate the conditions of the primitive early Earth? And if we do simulate them, what will happen to molecules of water, hydrogen, methane, etc.? Is particularly of interest was, may they, or could they, can they combine so that small monomers of molecules of life appear? And if you recall from Bio1 or from other courses, that living cells, living organisms have proteins, complex carbohydrates, lipids, nucleic acids. Right? They are polymers. And for them to form, a cell needs to have monomers. For example, nucleotides, small molecules which are monomers of. Then, the next question. Life requires large organic molecules, polymers. You probably know from, or remember from Biology 1 or from some other courses that cells consist of lipids, proteins, complex carbohydrates, as well as simple carbohydrates, and these nucleic acids, and these polymers must be formed again. They must be synthesized, and always when scientists ask about synthesis of something or making of more complex molecules from simpler molecules, they're always curious. So, what provided energy for this? Synthesis requires investment of energy. And at this point, there were several competing hypotheses, and one of them, the one that gained the most experimental support, was abiotic origin hypothesis, which implied that monomers link or form from polymer and form polymers without any enzymes. Remember from previous courses, enzymes, biological catalysts, and without other cellular equipment, without structures that generate energy. How come? What's still there? Some energy need to be invested. And one simple hypothesis was that when the liquid water, formed waves, this primitive, huge ocean that covered most of the surface of the Earth, experienced some storms or waves that contained monomers like nucleotides, amino acids, glycerol, glucose. They splashed on hot rock. And basically, the picture of the early Earth, 3.5 billion years ago, was ocean plus little islands of still hot rock. And when the splashing occurred, the heat was transformed into energy that was used for binding together, linking together the monomers and forming polymers. Okay, so the first step in the formation of life was probably making monomers of future molecules of life from compounds that traveled to us from other galaxies. The second step involved building of polymers from monomers using the heat of the Earth that was still not quite chilled. Now, yet another step, replication. See, if you just mix molecules of mixed proteins and lipids and complex carbohydrates, you will obtain some kind of soup which you can enclose it into some membrane, but it will not replicate. So the essence of life is life should beget life, right? Life should copy itself and reproduce. So what about that? What hypotheses exist about the first replicating structures during the forming of life. And the most popular hypothesis is that the first self-replicating structure during the forming of life was RNA. You see, the DNA, this classical genetic material, classical self-replicating molecule that forms complementary strands based on two original strands, etc. It needs enzymes to replicate. You might have learned in Biology 1 or in some other courses that, maybe anatomy, physiology, microbiology, that there are enzymes like helicase, DNA polymerase, etc. They are proteins, right? And the first structure probably was first self-replicating structure was simpler and less fastidious. It did not require protein enzymes. And RNA is such a structure. You see, RNA, some forms of RNA are biological catalysts. They're not proteins. They're not really enzymes. Sometimes they're called ribozymes. And they can accelerate the replication of RNA strands. And so for a while, what directly preceded forming of the cellular forms of life was what is sometimes called as RNA world, when there was this young planet, there was this primordial ocean, which contained tons of different inorganic and organic structures, including monomers of proteins and nucleic acids, including proteins and nucleic acids polymers and lipids, etc. But it also contained molecules of RNA which were copying themselves. And you see, nothing competed with them in copying themselves and replicating. So you can imagine that over millions of years, the primordial ocean was ripe with self-replicating RNAs. Then what happened next? First cells appeared. And it's interesting, how do we now envision the forming of first cells or pre-cells? Rather, their forming was amazingly simple. When you mix oil with water, which is of course impossible, but you can try, right? You have two phases. There is an aqueous phase or water down below, and liquid phase, fat, lipid, whatever, in the like forming a ceiling. And it is possible at certain conditions, and biophysicists showed it very well, but at certain conditions, lipids form circles and enclose the aqueous phase, enclose water with everything soluble in water, in that water. So a simple bubble forms, right? Bubbles like this. And these bubbles, again, primitive primordial ocean was ripe with all kinds of inorganic and organic molecules, including self-replicating RNAs. So these bubbles contained proteins, polypeptides, this slide says, and these bubbles also had RNA, which could replicate and used for synthesis of more of the polypeptides. Right? So these first pre-cells, they were very simple. The simplest living organisms now are far more complex than these pre-cells. But still, in what way they resembled real cells that we observe today? They had a membrane, the outer circle formed by lipids. And cytoplasm, which is this soup of various inorganic and organic molecules, including self-replicating RNA, dissolved in water inside. And the lipid membrane is not permeable for these solutes like salts or sugars or RNA, polypeptides, so they remain inside. They're not lost by the cell. And again, these pre-cells were back then, some 3 point something billion years ago, they were the only kind of cells or structures that could maintain their structure without losing, shrinking or swelling, without losing solutes, and able to build more protein and able to replicate. So they had no competition. So they multiplied to astronomical numbers, trillions, quadrillions over quadrillions. There was, at some point, there was a huge amount of them. And then, of course, there were some of these pre-cells that did better than others, were better adapted to the environment and survived, while other bubbles with proteins and other replicating RNA did not make it. And there were some changes in the RNA. Of course, the nucleic acids mutate. Again, you remember from Biology 1 that there are mutations in both in DNA and in RNA. So some changes occurred in the RNA and in the proteins. And then at some point DNA emerged. DNA, as you remember, RNA is normally single-stranded, but DNA is double-stranded, right? And it has some little differences in terms of nitrogenous bases with RNA, but it is a molecule that is much more stable than self-replicating RNAs. A DNA molecule, if you isolate DNA and put it in a plastic tube and forget this tube, or if it will fall on the table and you find it in like 5 years from the day when it was lost, this DNA will be still intact and functional, right? RNA is much more fastidious. RNA degrades very fast. So cells that evolved DNA were much more stable and more vigorous, survivable than pre-cells that had RNA only. Now, the first cell, this shows you a cell rather, that has a membrane and some kind of soup in it, cytoplasm, with self-replicating RNAs. Gradually, this evolved into a cell that had RNA and DNA, and eventually this mechanism of synthesizing proteins was developed when the original information about the structure of proteins is in the DNA, then DNA is transcribed into RNA, RNA is translated into polypeptides or proteins, and so cell becomes functional. It uses these proteins for various purposes, and it replicates. And this says that the first primitive membranes were most likely fatty lipids, so simply lipids, which spontaneously formed vesicles in water. If you remember the structure of a typical cell, uh, how, what, in what way are modern cells different from these pre-cells? What else do they have except lipid in their membrane? What do they have? Proteins. Like some proteins serve as protein channels, right? Ion channels, glucose transporters, etc. Other proteins embedded into the lipid membrane serve as enzymes or receptors, etc. And all this evolved very gradually, requiring billions of years of evolution. And we will have a separate cycle of lectures about evolution later in the course. Now let me switch to your lecture 3, prokaryotic diversity. A little bit of a jump, right? So from molecules to pre-cells to prokaryotes. What are prokaryotes? Uh, literally, prokaryote in Greek means before nuclei or before nucleus. So these are the cells which have no nucleus. That's their paramount features. Uh, they also are all unicellular, so one cell is one organism. Uh, they're smaller than typical nucleated cells or eukaryotic cells, and again, they have no nucleus. Now, it is hypothesized, and there is pretty strong evidence that pre-cells evolved first into prokaryotes, and then prokaryotes evolved into eukaryotes. So that's the reason we study prokaryotic cells first. Now, the entire life on Earth is classified into three very large groups, or called domains: archaea, bacteria, and eukarya. Archaea and bacteria are prokaryotic organisms, or prokaryotes. Eukarya are eukaryotic organisms, plants, animals, bears, birds, bees, humans, you and me. We all belong to the domain eukarya, but domains archaea and bacteria include only unicellular microorganisms, invisible without microscope, which are prokaryotic. Uh, just to summarize again, prokaryotes are the earliest organisms. They appeared hypothetically about 3.5 billion years ago. They're unicellular, they have no nucleus, and importantly, they do not have membranous or membrane-bound organelles. If you recall from Biology 1, you studied like typical cell, right? And your instructor probably told you stories about nucleus. Okay, then this slide shows you the structure of prekaryotic cells, or a prekaryotic cell. What do you see here? The cell, it happens to be a bacterial cell, has no nucleus, right? It's just the paramount feature of prokaryotes. It does have nucleic acids, though. It has DNA and RNA, and they are simply lying somewhere in the cytoplasm. They're not surrounded by nuclear membrane, so there is no nucleus. But the place where the DNA and the RNA are in prokaryotic cells is often called nucleoid. See, there is on the right-hand side of the picture, there is a pointer pointing at some space filled with double spiraled DNA and single spiraled RNA, and also some dots which symbolize emerging proteins and proteins attached to nucleic acids. So that is called nucleoid, but it's not an organelle. It's just again, it's a place where you can find the nucleic acids. Then there is cytoplasm, of course, all this soup of inorganic or organic molecules. There are some ribosomes. See, there's no synthesis of protein without ribosomes, so ribosomes have to be there. Note that there are no mitochondria. The structures that generate energy, generate ATP, if you recall from Bio1, they're in the so-called typical cell, a mitochondrion, or in plural, mitochondria, are responsible for making ATP. You don't see mitochondria here. So the structures that are needed to generate ATP are simply bound to the cellular membrane from the inside. Then you see the cellular membrane, right, cytoplasmic membrane surrounding this whole cell. Then outside of the cellular membrane, there is cell wall. It is pictured in green here, and note that green layer, which symbolizes the cell wall, is outside the cytoplasmic membrane or cell membrane. Cell membrane and cell wall have different structures. You recall from Bio1 or maybe from other courses that cellular membranes generally are lipids with attached or inserted, embedded proteins. A cell wall is not built like that. Cell walls in prokaryotic cells are actually built of large polymers, which are, by their chemical nature, they're complex carbohydrates. Now, the function of the cell wall, of the cellular membrane, as you probably know, is to regulate the transport. The membrane has channels which allow water and solutes to move out of the cell and in the cell. The movement of many solutes is regulated because channels that allow this movement are either closed or open. Water is not regulated. There are just water pores that are always open. But what regulates the movement of water either from outside into the cell or from inside the cell out is osmosis. Right? The gradient of solutes inside and outside of the cell, particularly salt and special proteins called albumins. These two factors are crucial for osmosis, so their concentrations regulate the movement of water. The function of the capsule, of the cell wall, green structure, is to protect prokaryotic cells from invasion or from harm. Right? Cell walls are mechanically stable, so it's due to cell walls it's hard to squash prokaryotic cells. Then cell walls also are important in not allowing some cells that are hostile to prokaryotes, like white blood cells inside the animal or human organism, to destroy prokaryotic cells. Then you see attachments, right, or appendages showing from the cell wall, or actually they begin in the cellular membrane and then they span the membrane, the wall, and also the capsule, if it exists. Capsule is an extension of the cell wall, which is built of polysaccharides. These appendages are of two kinds. There are short ones. On the left hand side of this picture, closer to the bottom, there is a word pili or pili, and the pointers show the shorter appendages. And there's usually one, sometimes two, long appendages. In this picture, it's one long appendage called flagella. Right? Pili or pili are structures that allow contact with the environment. They may be used for attachment by some prokaryotic cells. And flagella has a different function. Flagellum, actually, singular. Flagella is plural. Flagella are for movement. In many prokaryotic cells, these flagellas work like propellers. They move around kind of clockwise or counterclockwise and propel the cell forward or backward, or some complex movements of flagella can pilot the prokaryotic cell to the left or to the right. So it's an interesting and important organ of prokaryotic cells. Okay, let's move on. A few words about the diversity of prokaryotic organisms. There are two domains of prokaryotic organisms. We touched on the concept of domain earlier in the lecture. Bacteria and archaea. Bacteria and archaea are both unicellular, both prokaryotic, so they have no nucleus. One sentence literally about archaea, which distinguishes them from bacteria and actually from all other organisms that live on our planet. Archaea often live at extreme conditions, extreme hot, extreme cold, extreme salinity, etc. So you see here, the picture shows you the body of water which has bright blue and green spots. Those are thriving archaea. And note that this body of water is surrounded by literally boiling water vapors, which have the temperature of 100 degrees Celsius, right, or 220 degrees Fahrenheit. And at that temperature, some archaea, like these blue and green colored organisms, are very comfortable. It's kind of strange because we are used to the idea that cells have proteins. Proteins are sensitive to temperature, right? If the temperature grows, proteins denature. Like when you boil an egg, you see that the egg white changes from liquid to solid. That's because proteins in the egg white, albumins, are denaturing. But the thing about archaea is that their proteins may withstand very high temperature. They have a different way of denaturing. Just the high temperature would not denature them. On the other hand, some archaea live in the extreme cold. For example, if you go to Greenland, most of Greenland is covered by the ice shield. And if you drill like half a mile in the Arctic ice shield, the temperature there is well, way below freezing, maybe minus 100 Fahrenheit or something like this. And there is life there, and this life is reproducing archaea. Then there are archaea that are amazingly resistant to salt. for centuries and centuries, people used salty water, brine, right, to conserve vegetables, to conserve other food products, protecting them from being infested with bacteria mostly or fungi. And indeed, bacteria or fungi do not like high concentration of salt, sodium, sodium chloride. Uh, but there are some archaea that actually like it when it's very salty. You know, the saltiest body of water on Earth, it's the Dead Sea. The Dead Sea in the Middle East, it's in the border between Israel and Jordan. Large body of water, and it has so much salt that uh, even a person who does not know how to swim can easily kind of swim in the Dead Sea because the water is so salty that it carries a person's body. The body does not drown, does not immerse. The salt does not allow that to happen. And there is life in these extremely salty waters of the Dead Sea. And again, this life is archaea, unicellular prokaryotic organisms, sort of similar to bacteria, but on the other hand, different. There are some differences in the nucleotide sequences and molecular genetics, so they are considered two different domains. But again, to summarize, what you need to know about archaea is that they are extremophiles. They love extreme conditions, and they live at conditions that no other form of life on Earth can live. About significance of prokaryotes, particularly bacteria. About 5% of known bacteria may cause infectious diseases. That's the truth that has been there for centuries, beginning from Pasteur and Lister and other scientists and medical doctors. We know that there are some pretty nasty diseases that are caused by bacteria. For example, this is on the left side is a person ill with anthrax. It's a horrible disease which is caused by a bacteria, Bacillus anthracis. This bacterium produces a venom, a toxin, which causes swelling of the brain and necrotizing or dying of the skin and muscle. So it's like combination between gangrene and brain swelling. Very difficult to treat. These patients need massive doses of antibiotics, and even with these massive doses of antibiotics, mortality of anthrax is still pretty high. As you probably know, anthrax was used by bioterrorists. The dried form of Bacillus anthracis, the so-called endospore of Bacillus anthracis, was put in envelopes and mailed as U.S. mail to people whom the bioterrorists wanted to kill. But unfortunately, some absolutely innocent service people in the U.S. mail who handled these packages and envelopes got infected, got real anthrax. They were cured, but it was a big problem. And in the middle, you see a little child, probably newborn or maybe a few weeks of age, and this child has listeriosis. It's an infectious disease caused by a bacterium, Listeria monocytogenes, it's called. And the infection occurs in the mother's womb. And you see in this child, the affected area is the navel, belly button, and the surrounding skin. It's pretty nasty disease. It's treatable, but uh, children who are infected in the uterus with listeria and develop listeriosis, sometimes they develop some defects. They have malformed skull, difficulties in speaking, and difficulties in moving. So it's a nasty disease. And so pregnant women must stay away from listeria as far as possible. Then on the right hand side, there is a face of a person who is like a mask, right? The person is ill with botulism. Again, it's a disease caused by bacteria. Uh, in fact, the toxin, the venom that is produced by these bacteria, Clostridium botulinum, directly causes the disease, not the bacterial cells themselves, but the toxin. And what this toxin does, it paralyzes the musculature, beginning from small muscles of the face. Usually the first muscles affected are the muscles that regulate the movements of eyeballs, then the muscles that allow you to smile, frown, etc. All of them are paralyzed, and so the face develops a mask-like appearance, like nothing moves on this face. And then the paralysis spreads to the entire body, and if the person is not given the antidote, the anti-venom, antitoxin, the person may die because the diaphragm, the major muscle that causes actually the influx of air into the lungs, can be paralyzed. So in this case, the person must be put on life support, ventilator as soon as possible, or otherwise this person dies. So this is about the bad side of prokaryotic cells, particularly bacteria. But note that only about 5% of known bacteria may cause infectious diseases. Uh, the remaining 95 are not, we say, non-pathogenic. They don't cause any disease. If they are in your body, they are either indifferent, you don't really know about them, you don't care about them, uh, or useful. We'll talk about useful bacteria in a little bit. So this is about useful bacteria. Uh, it says cyanobacteria produce oxygen. Cyanobacteria are bacteria that thrive in shallow seawater, and they form huge colonies. Cyano implies that they are like cyanus in Latin means blue. They are bluish or greenish. They may be found in also in swamps, in on the banks of rivers, forming peculiar greenish, bluish uh, spots, etc. The wonderful thing about these bacteria, they are not harmful, they don't cause any diseases, but they produce oxygen because they are photosynthetic. You probably, you were to some extent at least exposed to the ideas of photosynthesis in biology 1 or maybe in microbiology. The photosynthesis basically means utilizing the energy of the sun directly to produce ATP, and then utilizing this ATP to produce the food which the bacterium consumes. So these bacteria like this are also called autotrophs, right? They feed themselves. They don't need nutrients from outside. Uh, and during the photosynthesis, actually at the very early stage of photosynthesis, molecules of water split and oxygen is produced, and oxygen diffuses out from the cell to the atmosphere. This is of huge importance. Now, all photosynthetic organisms produce oxygen, and for a pretty long time, it was the general kind of consensus that it is so important to conserve, to protect forests because forests produce so much oxygen, which is actually true. You know, the rainforest in the Amazon River basin in northern part of South America really produces megatons of oxygen. But we now know that cyanobacteria produce even more oxygen. So the life on our planet as we know it, our own existence, we humans also rely on oxygen in the atmosphere. We have to thank cyanobacteria for being there for us, making oxygen for our consumption, for our well-being. Then another thing, see the sketch on the right-hand side of this picture, to the right from cyanobacteria, shows some plant growing, and there is a little ball attached to the root of this plant, and inside this ball there are prokaryotic cells, bacterial cells. Now we say that these bacteria associated with roots of certain plants, legumes, they are nitrogen fixing. What does it mean, nitrogen fixing? You see, 80% of the atmosphere is nitrogen, right? And every single organism living on Earth needs nitrogen because nitrogen is a part of nitrogenous bases, which are where? In the nucleotides, in the DNA, in the RNA, right? So getting some nitrogen is vitally important. If we don't get nitrogen, we cannot make nucleotides. But amazingly, we cannot get nitrogen from the atmosphere. These bacteria can. They get nitrogen directly that diffuses into the soil directly from the atmosphere. And then they convert this nitrogen into nitrogen-containing compounds like nitrites, nitrates, etc. And the plant which harbors this little ball with bacteria benefits from that. Right? It utilizes the nitrates and nitrates because it has some enzymes, some machinery in the plant cells that allows to synthesize nucleotides or use nitrates, nitrites in for some other purpose. Then the cattle eats these legumes. We eat these legumes, right? And the cattle gets the nitrites and nitrates from this legume, and when we eat a hamburger or a steak, we get nitrogen from the cattle, from the cow who ate the legume, which had nitrogen-fixing bacteria associated with it. So the existence, again, we owe our life to the nitrogen-fixing bacteria. So that's how useful they are. And third bullet point, many bacteria in the human intestine produce useful substances, vitamins. Just an example, you have, all of us have colonies of a bacterium called Escherichia coli, which lives in the colon, in the large intestine. and it produces a substance known as vitamin K. Vitamin K is a blood clotting factor. There is no blood clotting without vitamin K, right? We can get some vitamin A with diet when we eat our food, but there were some calculations that showed that our diet covers only about 20% of our need in vitamin K. The rest, 80%, comes from bacteria, from Escherichia coli that live in the colon. So again, very important. Without E. coli prospering, thriving in our colon, we would bleed to death. Because without vitamin K, there is no blood clotting. Now, bacterial ecology. It shows you, again, this perennial fight between the good and the bad. There are good bacteria, friendly bacteria, particularly in your intestine. This picture shows the inside of the small intestine with these microvilli, the appendages that increase the surface and this is beneficial for absorption of the nutrients from the inside of the intestine to the blood vessels, so that your organism is richly supplied by nutrients like glucose and amino acids, etc., by the blood flowing through your entire body. And in this small intestine, you see little green guys, right, which are marked here as friendly bacteria, good bacteria. Some of them, this is not a detailed microbiology course, but I'll just note that the first one in the list, L. acidophilus, and it's but bacteria, basically it's bacterium that causes fermentation of milk and dairy products, converting their materials into lactic acid. Lactic acid is something that muscle cells especially need. And generally all cells need it to some extent, need this lactic acid. So these are good bacteria, these are friendly bacteria. And then on the right part there is this black spot, and it shows you unfriendly bacteria. They resemble like some poisonous mushrooms or something, or some worms that harm your body. It says Candida albicans. Actually, Candida albicans is not a bacterium, it is a fungus, and like all fungi, it is eukaryotic, so we will not talk about them here, but I'll just mention there are some bacteria which are very unfriendly, bacteria that cause, for example, salmonellosis or dysentery or plague even. They can also thrive in the same place where these fermenters of lactic acid grow. And there's always a kind of fight between them. And the one practical thing about, or practical implication from this knowledge about bacterial ecology, when you prescribe antibiotics to a patient, be very careful. Prescribing strong antibiotics at large doses for a very long time may destroy this fine balance between friendly and unfriendly bacteria. It may cause massive death of these lactic acid fermenters and inability of the organism to ferment lactose or ferment dairy products into lactose. So antibiotics may be quite dangerous. They will destroy the unfriendly bacteria, but they also may destroy friendly bacteria. And so the balance in the intestine will be shifted, and sometimes it's even worse than the problem that actually prompted the prescription of the antibiotics. Total annihilation of bacteria from the small intestine kind of gives the green light to other forms of life which are not bacteria and not sensitive to antibiotics. For example, fungi, Candida albicans, the same, the one that is mentioned here in the text. So and candidiosis, or the proliferation of fungus in the intestine, may be quite unpleasant, and sometimes doctors actually regret that they prescribed so much antibiotic in such a strong, big dose and for such a long time that it just wiped out the entire host of bacteria and gave the advantage to fungus. That should not happen. Prescribe antibiotics at reasonable doses for reasonable time. If you need to prescribe them for a very long time, sometimes, in patients with chronic diseases, you need to keep them on antibiotics for months and years. But then in parallel, give them antifungal preparations like nystatin. That is a huge thing that really keeps alive a lot of patients with severe infectious diseases. One important point in this lecture, particularly about bacterial cells. Bacteria do not reproduce sexually. They reproduce simply by binary fission, meaning they split into two parts. So a colony of bacteria, it's actually a clone. All the children are exactly the same as the parental bacterial cell, children, grandchildren, great-grandchildren, etc. But back in the 1950s, what microbiologists noticed that bacteria form kind of pairs, right? And it sort of reminds of sexual activity between two two persons in a couple. But it's of course it's not sexual reproduction, but it is exchange in the DNA. The big part of bacterial DNA is it's on the left-hand side of this sketch. There is a large blue circle in a bacterial cell. That's the bulk of the bacterial DNA. Sometimes it is called chromosome, bacterial chromosome. It's not quite correct. Bacteria do not have real chromosomes, but it's the bulk of the DNA. But see, underneath this large blue circle, there is also a smaller circle which is painted bright red. And in the middle, you see what happens? The bacteria on the left gives its little circle of DNA to the bacteria on the right. This circle, little circle, small circular structure, which is bacterial DNA, it's outside of the bulk of the DNA. It can be separated physically from the bulk of the DNA, and microbiologists called it plasmid, singular plasmid or plural plasmids. So bacteria can exchange plasmids, just like in this picture it shows that the left bacteria gives its plasmid to the right bacteria. Uh, it is possible, the other way around is possible. The bacteria on the right could have had the plasmid and transfer it to the bacteria on the left. What is the meaning of this exchange of DNA? The thing is that like we just stated that bacterial population or colony is a clone, right? All individual cells are identical, but not quite identical. See, some of them have plasmids, and others may not have plasmids. And plasmids are DNA, so plasmids do have some genes, and not having a certain plasmid may be detrimental for bacteria. It's in the 1960s, 70s, there was really an avalanche of research showing that genes that confer resistance to antibiotics usually are associated with plasmids, not with the bulk of the bacterial DNA, but with plasmids. So the harboring this plasmid with genes that make a bacterium resistant to antibiotics, it's very important. If uh, if, imagine that like 10 million bacterial cells are exposed to an antibiotic. There is a chance that at least in some of these bacteria, say in 500 cells out of 10,000, out of 10 millions, there is this magical plasmid that confers antibiotic resistance. Then the most members of this colony, what's that? 9,500,000 cells, right? They die, but the remaining 500 are now uh, getting a lot more nutrients, right? They are no longer competing with non-plasmids containing bacteria. So they replicate, they eat well, and they replicate fast, and in some uh, maybe 2, 3, 4, 5 days, there will be a colossal amount of bacteria that are antibiotic resistant. They will grow to the original 10 million, and then they will make 20 million, 40 million, 80 million, etc., billions of individual cells. This makes, again, poses some limitation on the usage of antibiotics. Uh, Unfortunately, many physicians are kind of relying on the latest fad. Like there is an antibiotic which everyone knows, everyone prescribes it, etc. It is known to be very powerful. It destroys bacteria that cause some nasty diseases. But what if this antibiotic is not acting on those bacterial cells that harbor this magical antibiotic resistance conferring plasmid? So then this antibiotic is useless, totally useless. No matter what they write about it in advertisement kind of literature by pharmaceutical companies, don't waste a patient's money, and especially the precious time treating a patient with antibiotics that has no effect on the bacterial cells. So better, in general microbiology, we have a special lab testing resistance and sensitivity to antibiotics. There is a special test called Kirby-Bauer antibiotic sensitivity resistance test. It's pretty simple, actually. It can be done even in our tutorial laboratory, so it of course can be done in any laboratory associated with a hospital or inpatient unit. But sometimes, there's double pressure. One pressure is from the industry, from big pharma, which just sort of insist that doctors use this antibiotic. And the other pressure is, especially in pediatrics, it's the pressure from parents. Kirby-Bauer test takes 48 hours, so the results are visible in 2 days. And a furious mom can, in these 2 days, can raise hell and say the doctors are not doing anything. They're not. It's obviously an infection, and they're not giving an antibiotic to my child. But the thing is that it may be better to wait these 48 hours and then prescribe the antibiotic that works than to prescribe antibiotic from the very start, seeing that it doesn't work and still giving it to the patient. That would be kind of dangerous. Okay, and I think this is the end of our lecture about prokaryotes and their diversity. Stay tuned.